through a titanium heat exchanger where it flows countercurrent to adjusted quantities of hot or cold fresh water. And in this way, the temperature is tightly manipulated. Temperature is typically set at a delta from the incoming reef seawater in order to maintain the dial variability. Light intensity, spectra, nutrients, feeding and flow are also all easily manipulated on an individual aquaria level. Temperature, pH and dissolved oxygen are all monitored quasi-continuously by this robot. Additional probes such as a monitoring pram or an underwater camera can be added as required. The data is reported in real time to an online software through which the researcher sets the system to follow user-defined values. Additionally, multiple sensors are in place to raise an email or text message alert when there are faults or if environmental parameters breach the defined range limits. Predominantly, we work with reef-building corals in this system, but we have also experimented with soft corals, sea urchins, fish, seagrasses, and for many months at a time. In addition, hard and soft corals in the system have maintained reproductive synchronicity with the field populations. Such systems greatly enhance our ability to run ecologically relevant experiments and to draw conclusions from them that fit with our changing oceans. We welcome your contact for further discussion, so please get in contact via our Twitter page, email or website. Thank you.
Okay. Good. Okay, I can get rid of this. And now I can stop the screen sharing. There is one chat I just wanted to see. Oh, okay. Okay, that was it. Okay, so I think in this past sort of 20 minutes or so, you've had a nice overview of what the area looks like, why we're there, what we do, some things about what we can do. We get requests sometimes from companies where the researchers are involved in joint projects. It just depends. Some companies, for example, were interested in the octopus and trying to study camouflage. Can we use this for other reasons? Or dolphins, for example, were found to brush themselves, rub themselves on some coral reefs. And the question was why? And it was found recently that the corals may have some kind of enzyme, whatever, that may help the dolphin overcome some kind of infection or a scratch or something that's bothering them. So basically, or for example, the corals ultraviolet, trying to understand how we could make a better sunscreen to overcome environmental stress, which can be used by people, which is more environmental friendly than the existing sunscreens. We have quite a few problems in the lab because of the tourism. Everyone's going in with sunscreen, and not too many products that are considered, say, environmental friendly. So our companies are coming in and interested to find out what they can do, not just here, but it's something globally. So perhaps, with having said that, perhaps open the floor to questions or comments that people may have.
Yes, thank you, Simon. Any questions about the institute at ELAT? I can witness that it's a fantastic place, very good conditions for research, and it's amazing place. And yeah, I think that one thing, Simon, I just wondering, the decision to have the institute, like you highlighted, did it come from the universities themselves, or is it a government initiative or some outside initiative?
Well, what happened is originally, the whole institute is owned by the Hebrew University of Jerusalem, and for about 20 years it was run only by the university. When they wanted to build labs and a campus, the government stepped in and said, well, we don't want to just fund one university. Mm-hmm. And every university will say that. So the initiative was of our university establishing itself, and then the demand, okay, for research and the understanding that this was a very, very valuable tourism place for Israel. And therefore, it needed to have the corals protected. Studies, less developments would destroy the golden egg. And so the universities, they said what it did, the government was just made us an offer we couldn't refuse. Just saying, we'll basically fund most of your work. Your university, your respective institution will still have to fund positions, etc., some things, but we'll support the campus. We'll give you an annual budget, which is reasonable to operate in. And the rest you get from research grants, government infrastructure grants. Everything's done competitively. In other words, even to set up such a campus, they looked at other topics, science topics in Israel, might be electronics, physics, chemistry, and should we invest our money in something else? So luckily at that time, this was a key area, and the money went to establishing this campus. Still get support, as we said, by the government, as well as dedicated calls for, let's say, marine infrastructure, like how you get the new building that we have, for example, which we just saw at the end, or the remote underwater vehicle, or other major pieces of equipment. We have an Israel Science Foundation, which will fund also the researchers there to establish, let's say, of special labs that you couldn't normally establish with a research grant. And then it's shared, or part of it is shared. And the idea, you're basically building a community who shares things as far as possible. And that's what makes it a bit unique, since you have this huge number of people, resources of people and their skills that you can call upon.
Yeah. And I think, do you have also, I guess there must be other examples in other areas of science where, like in the desert or whatever, where you get this, different institutes together, or is this just a single case?
This is very unusual. It's the only one in Israel. I've been working for a desert center, also previously terrestrial ecology, for many years in the Negev desert, north, almost close to Eilat, running up to like almost Beersheba, which is like the northern part of the desert. And there is a desert center at Ben Gurion University, which has also its own campus. But here, everyone is, there's no, let's say, shared campus. It might be a good idea, but for whatever reason at the moment it isn't. And it's very hard, I mean, for any country to share facilities, because everyone's going to get jealous, who's getting more, who's getting priority. And we have a board which represents every university. Everyone gets along, everyone does things reasonably. The government is reasonable in their funding. And then it's normal competition, people applying for grants, which is individual. So in this case, I think to answer your question, it is unique in Israel. And I don't know actually too many places around the world, if any, who also have a similar setup, where people are sharing all their facilities. Everyone wants their facility. In our case, it's such a short shoreline, it's only 11 kilometers from the Eilat city to the Egyptian border, that there isn't room anyways to set up facilities.
Yeah, that's the other thing. It's you minimize impact. It's a special place, so if everyone wanted to have a little bit of it, it would be disastrous. But but I think it's very good, and certainly there are big advantages in sharing not just expertise, but this common equipment and facilities and so on. And it's excellent. Everyone that has been there speaks well about it, and I can witness, I guess, as well. Well, is anyone else would like to ask a question? Otherwise, we'll say thank you, Simon. We'll wait a couple of minutes just to get near to the normal time for the next talk. And then we'll continue with Stazione Zoologica.
Okay. I'd like to thank you for having organized this. This is a very hard workshop to try to do over 10 days and to coordinate, etc. So more power, Adelino, to you and your elbows, your typing elbows. Okay.
Yeah, I think it's just going to work. We will have to you know, maybe every 2 years or something, maybe a bit better organized, you know, because it has been very much based on a small group. But I think it's, you know, it's a good, there's a lot of people that come through, you know, maybe in each session, you may not have more than 30, 40, sometimes 50 people, but overall, there are a lot of people that come to this conference, and that means that, you know, they get in touch with the concept of the infrastructures, and they are really potential users, many of them, particularly for PhD students and other students. So I think this is, you know, last year, of course, people were more into the online. This year, there is not, you know, everyone is more wanting to be offline, but still, we have more than 200, about 250 people registered at the moment, and I think during the week there'll be more people.
Okay.
Okay, so thank you, Simon. Now it's the Stazione Zoologica Anton Dohrn in Naples, and it's going to talk about a particular service that they have, which is the taxonomic services. And this is, you know, it'll be Iole Di Capua, which I think is probably managing this service, that will be talking. So please go ahead, Iole.
You like the presentation?
Yeah, that's fine.
Okay, perfect. Good morning, everyone. And today I would like to introduce the only service of our institute, Marine Organism Taxonomy Service, that we call it MOTAX. MOTAX is a facility of research infrastructure, marine biological resource department, and of Stazione Zoologica. And Stazione Zoologica is the headquarters of European Marine Biological Resource Center of Italy. And EMBRC Italy coordinates different 16 members, 8 universities, 4 public research institutes, 1 national agency, 1 national technological cluster, 1 inter-university consortium, and 1 zooprophylactic institute. And our service is a distributed taxonomy reference platform for EMBRC, and the call for Assemble Plus are open now. Go back to Stazione Zoologica Anton Dohrn. Stazione Zoologica was founded by Anton Dohrn in 1872, and this year so we're celebrating the 140 years of its foundation. And is placed in Naples, in front of the wonderful Gulf of Naples. And since its foundations, the Stazione Zoologica have a traditional knowledge in taxonomy, in particular on morphological taxonomy. And for these reasons, during the years, have passed more than 415 taxonomies in our institutes, because Anton Dohrn invented the bench system that in Italian we call the banchi di lavoro, and provide the Stazione Zoologica as a service, because for a fee, he hosted the scientists, mainly taxonomy, for research. And for these reasons, we have studied in our institute more than 20 phyla of marine organisms, and we have a very big zoological collection, and this website is completely dedicated to this collection. It's possible to look the preserved material in 3D view, and we have also many materials, type materials of the first identification of taxonomic identification of specimens, mainly from Gulf of Naples. And we have more than 40 monographers and over the 2,500 systematic publications that we have collected in the past and have already used for studies of morphological taxonomy of these organisms, like stomatopods, copepods, acantarians, and so on. And all for these reasons, and also for the impediment in taxonomy that we're living in the last 2 decades, was born the idea to create a dedicated service merging the traditional expertise in morphological taxonomy, alpha taxonomy, with new molecular and morphological approach tools. And in MOTAX, we provide, in fact, this expertise. And our mission is also to support scientific activities in framework of ecological, biological, and phylogeny studies. But our service is devoted mainly to train researchers and technicians in integrated taxonomy, and to share and sharing experience and expertise in integrated taxonomy. And for example, I will show you as we provide all activities from sample sequences at the Stazione Zoologica. We go at sea to sample of our sample in the Gulf of Naples and in the adjacent areas. We come in the lab to perform morphological identification, and also we are able to perform molecular taxonomy of these organisms. Our course facilities is mainly devoted to phytoplankton. We have 3 senior experts. The head of our facilities, Diana Sarno, Isabella Perkov, and Eleonora Scalco. And we have other research fellows that we help us in many monitoring and research programs. For zooplankton, I am myself the expert, senior expert of Stazione Zoologica, and I'm collaborating with other research fellows also to address different research programs. And finally, we cover the expertise of benthos. We have 2 main experts, Paolo Fasciglione and Rosanna Guglielmo, and other young scientists that help us in monitoring program mainly. But we have also a very well-structured network of Italian taxonomists that cover different expertise of different taxa, of marine taxa, that will help us in, for example, identification. And I will show you very briefly only few activities that we have in our portfolio as MOTAX. For example, we have able to identify at the species level phytoplankton, zooplankton, and benthos. We are also to done the biomass measurement using standard operation procedures that we are already prepared in our service. And we are also able to sort the target species or target specimen that you need. We are able to perform isolation and strain cultivation, for example, of phytoplankton species. And also we are able to address molecular characterization, for example, of different gene targets if you need for molecular taxonomy. And for sampling activities, we have 2 main big research vessels, Victoria and Hippocampus, and we are supported of another facility of our department of infrastructure of marine research. We able to perform morphological analysis under stereo microscope or using the lighted or inverted microscope. And if you need to undergo deeper in the morphological identification of a specimen, we are also supported by Advanced Microscopy Center to address scanning electron microscope, environmental electron microscope, or transmission electron microscope, Obviously, all these activities are supported and the preparation of the samples are supported by our experts. And also to be able to perform molecular identification and sequences of specimens. We have extracted DNA from a single specimens. We are able to do PCR chain reaction of target genes, and we have standard sequences to have sequences of specimens supported by molecular sequences and analysis center. And I want to show you some example of the user stories of Assemble that we have in the last years after pandemic situation. For phytoplankton, we hosted, for example, scientists mainly focused on to isolate strains of Spezionicea species. And another from Barcelona to perform morphological characterization of a new species of Heterocapsa. And we also host for staff exchange in framework of Assemble Plus, Cecirad Menandez from Scottish Association of Marine Science to address advanced taxonomy on marine diatoms. About example of zooplankton, last years I collaborated with Tamara from Israel Oceanographic Limnology Research to identify species of marine planktonic copepods from a historical site in Israel, and to compare her specimens with the specimens from the Gulf of Naples. And for the next July, we will host a young scientist come from Brazil, Erika Becker, to perform morphological and molecular identification of copepods from subtropical waters. About benthos, we host 2 scientists from Spain that are interested to identify meiofauna species. And another important activities that we have organized in motor services are the advanced courses. We, in the past, for phytoplankton, have organized more than 8 courses. And for the future, we have planned another course that we unfortunately have postponed for COVID. And all of courses are supported by EMBRC, and the cost of the fee were supported by EMBRC. For zooplankton, in the past we organized 1 advanced zooplankton courses completely dedicated on molecular and morphological taxonomy of marine copepods, mainly to different families, and we planned another course for the next years. And for benthos, we are going organizing a course on completely dedicated on integrated taxonomy of polychaetes in the next October, and the deadline is closed the last week. And another course is organizing for the next September, and this course is more practice, and we go in the field to observe and monitoring Posidonia oceanica in the Gulf of Salerno. And thank you for your attention, and we are waiting at Stazione Zoologica to perform your work in collaboration with Motex. The contacts are of Diana, if you want to other questions on our.
Thank you, Iole. Any questions about this, about the Stazione and the services, in particular these taxonomic services?
Oh, sorry.
No.
Maybe
How many? So come on, Simon. Sorry.
What is the cost of the service? If someone has a species, they want you to identify them, for example, what would that be? Or is it a free service, or is it a some procedure that you must do?
We have the costs are supported by EMBRC or by Assemble Plus if you need an HS, but we have a cost for each of these activities that we also able to have.
Is there a table?
Yeah.
Is there
as a
table
service for example, for industrial or for other activities, monitoring activities, and so on. In firm,
you have a table of the prices?
Yes,
we have
a table of prices of our portfolio of each activities, but in general, we have this cost for commitment, external commitment, not for researcher activities or for collaboration.
Mm-hmm. Okay, and do you get many people requesting that kind of service? Yes,
because our expertise is very particular, because it's not easy to identify at species level zooplankton, phytoplankton, and benthos, and there are few experts in Europe that are involved in this field.
Okay. Any other questions? No? Oh, well, in that case, thank you very much, Iole.
Thank you.
Good afternoon, everyone. Welcome to the Assemble Plus conference. This afternoon, we're going to have 3 talks, and these 3 talks will start now. The first talk will be by Stojmir Kolarevic of the University of Belgrade in Serbia. And he is going to talk about single and combined potential of polystyrene microparticles and fluoxetine in the induction of DNA damage in hemocytes of Mediterranean muscle. So thank you very much, Stojmir, for accepting to participate. And please go ahead.
Good day, everybody.
I think the connection is very slow. You disappeared, I'm afraid. It froze. It froze. It's thrown out. Let's see if he can come in again. I think he's having difficulties with his internet connection. No.
Sorry, the Zoom is kicking me out for some reason.
I think it's probably the speed is too slow, I think, the problem. But let's try again. Now you can see it well.
Okay, sorry again. Have you, I know how much have you hear me? So
we are hearing fine, and now it's okay the way you are presenting it.
Okay, thanks. So I'm coming from the University of Belgrade, from the Department of Hydroecology and Water Protection of the Institute for Biological Research. Together with Dr. Margareta Kračun-Kolarevic, we applied for the transnational access in 2019, and we have visited Marine Biology Station in Piran. At home, we are mainly focused on freshwater ecosystems, but we wanted to combine the knowledge that we have from freshwater ecosystems with the knowledge that a colleague from Marine Biology Station Piran in Slovenia have. So in 2019, we spent one lovely month in the laboratory of Dr. Andrea Ramšak and trying to carry out the proposal that we had at that time. I have to say that Assemble Plus was good kickoff, as after this project, we continued the cooperation with colleagues from Slovenia, and we also established trilateral cooperation with the colleagues from Montenegro. And after that, we got 2 bilateral projects which are still lasting. So today, I will present some of the data that we obtained during the Assemble project, but also from these bilateral projects. So you're probably familiar with this type of emerging pollution in aquatic ecosystems. So these are particles made of different materials such as polyethylene, polystyrene, polyvinyl chloride, which are less than 5 millimeters in diameters and have become the major concern present in ecosystems worldwide. Numerous responses are triggered in organisms such as inflammation, destabilization of lysosomal membranes, genotoxic effects, impaired reproduction, and imbalance of hormone synthesis. But the properties that we were really interested of microplastics are the vector effect that microplastic can have. So due to their hydrophobic properties and relatively large surface area, microplastics can have a so-called vector effect, acting as a carrier of hydrophobic pollutants in the water and facilitating their transfer to organisms through the food chain. So many authors have described this as these potential hazards of such relationship as the Trojan horse effect mechanisms, where the absorption of hydrophobic pollutants to microplastic particles can enhance or multiply their accumulations in the organisms. Such pollutants is fluorantane. It can be metabolized by aquatic organisms and may generate reactive oxygen species and form adducts, which can exert both ecotoxic and genotoxic effects if antioxidant defenses are overcome by pro-oxidant forces. And that's why the fluorantane was our major pick, because we wanted to study this genotoxic effect. And in combination of polyethylene and polystyrene, microplastic had significant effects on its accumulation and oxidative stress response in the muscles. But to our knowledge, there was only scarce data about its genotoxic potential when it's combined to microplastic particles. So the major goal of our study was to investigate the possible vector effect in the exposure of Mediterranean mussels to polystyrene microplasticles with absorbed fluorantane by using the multi-biomarker approach. During our visit, the major focus was placed on genotoxicological endpoints. As I said, since to our knowledge, there is no literature data on genotoxicity of polystyrene particles alone or with absorbed fluorantane in these mussels. And additionally, we investigated neurotoxicity by acetylcholine esterase activity measured in gills of experimental mussels, while GST was assessed in gills and hepatopancreas, as these enzymes are used for biotransformation and excretion of lipophilic compounds such as PHAs. Afterwards, with colleagues from Montenegro, we wanted to investigate whether the Trojan horse mechanism effect can be seen in the change of heart rate patterns within the exposure of mussels to polystyrene particles, fluorantane, or particles with absorbed fluorantane. So our study design looked like this. So we have collected mussels from a pristine site in Slovenia. We acclimatized them to laboratory conditions. Then we separate them in 4 groups. These were a control group exposed to polystyrene only, group exposed to fluorantane, and group exposed to polystyrene with absorbed fluorantane. And we have monitored the effect of this exposure for 96 hours. So we set up checkpoints at each 24 hours where we collected sample of tissues. These were hemolymph, hepatopancreas, and gills. And we performed comet assay and micronucleus assay on hemolymph, while GST and ACHE were assessed in gills or hepatopancreas. Afterwards, colleagues from Montenegro performed separate experiments where they exposed the same groups to the same type of pollutants and monitored their heart rate patterns for 96 hours. So, as I said, our major focus is DNA damage. We performed COMET assay. I don't know how familiar are with this assay. It is quite sensitive technique for detection of DNA damage. The name COMET assay is gained from these so-called comet-like shapes which damaged nuclei have. So on these images, you can see undamaged cell nuclei and damaged cell nuclei. So when you collect hemolymph, then you are mixing with agaroses, you're placing it on the slides, then you're performing lysis. After lysis, you're doing the naturation to unwind the DNA helix. Then you're doing electrophoresis. All the DNA which is damaged and break into fragments go to the tail of the comets. After electrophoresis, there is neutralization, then staining, and the last step is scoring on under the fluorescence. And we are using COMET Assay 4 software, which is used for quantification of damage. But the parameter that we are generally using is tail intensity, which is basically the percentage of DNA which is in the tail of the comets. What I must say now is that the type of damage that you detect with the COMET assay can have different fates. It can be either repaired, or if the cell is too damaged, then it can go to apoptosis, or if it's not repaired and if its cell does go into apoptosis, then it becomes permanent damage, which is detected by micronucleus assay. That's why we also perform the micronucleus assay, but only at 96 hours after the whole exposure. So these are the results obtained after 24, 48, 72, and 96 hours. And what we have noticed is that fluorantin, as indicated in black here, reached the top after 48 hours of exposure, and then it started gradually declining. But on the other hand, polystyrene with fluorantin was significantly lower at both times, but at 72 hours of exposure, it reached DNA damage, it reached its maximum, and then start to decline, while the polystyrene alone only had some evident effect after 96 hours of exposure. When we check the frequency of micronuclei, we get the significantly higher frequency of micronuclei in group exposed to fluorantin in comparison to polystyrene and fluorantin together. For GST and ACHE, we haven't observed some strict trend line in variation of activity. The only significant effect for acetylcholinesterase was noticed after 96 hours, where the groups exposed to the polystyrene with fluorantin had significantly lower activity of this enzyme, while for GST, we have observed statistically significant difference only after 24 hours of exposure. Still, we have observed some variations, and we are still looking further into this. And finally, heart rate. So the cardiac activity recording of muscles was conducted by non-invasive laser fiber optic method. Specific region of the heart was exposed to the infrared light beam, as you can see on image above. So the reflected light containing the information about periodical changes in heart volumes was detected by small optical sensors glued to the shell about the heart area. So after this reading, which is carried out every 10 seconds, you are getting this kind of image. Considering that we had a large amount of the data, we converted this data in parameter called DHR, or deviation in heart rate, which represents the average difference in heart rate response relative to the pre-treatment period. And this was used as indication of the stress level. So how it looks basically, this C1 on this image for control is the period before the experiment started. It is first few hours before we started with the exposure. Then this time 1, time 2, time 3, time 4 represents first, second, third, or fourth day of exposure. And the C2 period is the deviations in heart rate, which are measured after the treatment period. So after the treatment, we change the water and then see if the heart rate patterns are back to normal or they are still elevated. So once again, for fluorantin, we have observed during the first day the highest deviation in heart rate, indicated that the muscles were under the high stress this first day, and then it declined and stayed at some similar level till the end of the exposure. But for the polystyrene with absorbed fluorantin, we observed different patterns. We observed gradual increase in the deviations of heart rate, which reached its top after 96 hours of exposure, the last day of exposure, showing different trend, once again going in the favor of this Trojan horse effect that we wanted to investigate. Finally, we integrated all the biomarkers that we measured by integrated biomarker response. We used version 2. And again, you can see on this chart on the left that for fluorantin, the highest response of measured biomarkers was seen after 24 hours of exposure, and you have this gradual decrease. But on the contrary, for the group exposed to combination of fluorantin and absorbed to polystyrene, you can see the gradual increase in response in the first 3 days and then decrease after 4 days. Here on the figure on the right, you can see the relative contribution of each biomarker that we monitored to observe patterns. So here on the image, you can see that heart rate was the most contributing to the surface of IBR diagram in the group exposed to fluorantin, and you can see decrease of it in the next days. But at the same time, you can see the increase of the COMET assay after 48 hours. But for group exposed to polystyrene with fluorantin, this indicated in yellow here, you can see that you have gradual increase in COMET assay by the 3rd day, and you have constant increase in the heart rate patterns during all 4 days. Again, going in favor that muscle have a postponed response when the fluorantin is attached to the polystyrene. So this is only part of the data that we already processed. We have additional data that we are still processing for other biomarkers. But this is it for now. I would like to thank you for your attention. And again, I like to thank Assemble Plus for supporting our research and giving us opportunity for transnational access. Thank you.
Thank you, Stoimir. Anyone would like to ask a question? I have a couple of questions. So the one is about the heart rate.
Yep.
The patterns seem to be quite different.
Yes.
What would that mean? I mean, why sometimes it goes up very quickly and then decreases. Other times, like in the last, where you have the two compounds, it was a progressive, like why should they be different, do you think?
Yeah. If you look at the data that we observed, that we gained only for polystyrene, no significant difference in heart rate patterns. So we assume that when fluorantin particles, muscles do not recognize it, they do not have stress from it, and they have stress afterwards when the fluorantin is detached from the particles. And this is the process which needs probably a few days, and that's why we're getting the highest response after 4 days of exposure. On the other hand, fluorantin is directly absorbed by the muscles during the first day of exposure. We got the highest response, and then we have a kind of either adaptation to its presence or a complete accumulation of fluorantin.
Yeah, I was, when you showed those kind of, those graphs, the last graphs you showed, it seemed that after 96 hours, the area occupied by the various parameters is smaller, which suggests some kind of acclimation.
Yeah, there should be, because this is experiment carried out in static conditions. So we assume that there was acclimation after these 4 days.
Mm-hmm. Okay, very good. So that's I'm glad that it has worked. It seems as if we get a lot of data.
Yeah.
Okay, thank you very much. Oh, there's a, what's that? Let me check here. Okay, just a comment. If there are no more questions, thank you very much, Stoimir, and I hope, well, anyway, we expect to continue to have the possibility of offering transnational access, you know, later on, and later in the year, we should know more about it, but certainly we will communicate. EMBRC is having his own program, and also there's some partnership with Aqua Excel, so that's more aquaculture, and there are several opportunities coming up of funding from the European Commission. So some of it will come next year. You know, in practice, it will come next year. But I think we'll continue to provide these opportunities. Thank you.
Thank you, and again, my apologies for these technical issues. I'm very sorry for it.
No, but that's part of the online. The next talk is by Ana Filipa Lopes. She's from MARE ISPA. ISPA is the Institute for Psychology Sciences and Life Sciences. And she's going to talk about fish behavior and physiology under rapid ocean warming effects. So please, Anna, go ahead. Let's take the... You can share now.
Okay. Okay. Just a minute. Okay, I think it's working, right?
That's fine. Thank you.
Okay, cool. So hello, everyone. Thank you so much to Assemble and to Professor Adelino Canário for inviting me to talk today. And also thank you for the ones of you that are watching this. So the work I'm presenting here, it's part of my PhD thesis that I'm doing at ISPA, as it was said. I'm also a guest PhD student at the University of Gothenburg. And my PhD is it's a scholarship done through the FCT, and it's also part of a project called NextGen. And of course, it has the financial support. It had the financial support of Assemble. Okay, so where did I do this experiment, this work? I went to Sweden, to the Kristineberg Marine Research Station here, and I went and did some experiments. The Kristineberg Marine Station is in the Gullmarn Fjord in Sweden. It's a very beautiful place, as you can see. And we chose this location because of the characteristics of the fjord and the species we wanted to study that inhabits the fjord. The species is the two-spotted goby. The two-spotted goby is a semi-pelagical fish that distributes alongside the European coast. So the south it's Portugal, and the north it's Norway. And there is a population on the fjord between Norway and Sweden. And this is a really nice species to study because it has a small life cycle. And it's also well maintained in captivity, so we can use it as a model to test different behaviors and physiology. And our aim our goal was to address the effects of ocean warming on behavior and physiology of this population. So we had 3 temperatures, 15 degrees, which is the temperature, the environmental temperature for the month we were there doing this experiment. So it's the control temperature. We were there in June last year. And then we had 18 degrees, which is an elevated temperature, but within the fish range of variability. And then we had 21 degrees. There is a temperature that's outside the fish's range of variability. And we wanted to see how fish responded in their escape response, their feeding rates, and their routine metabolic rate. And so how did we do this? So we went on the fjord, we snorkel on the fjord to catch the fish. We caught them by with hand dipped nets. And then we brought it immediately to the lab, where they stayed in a quarantine aquarium for a week to just acclimatize to the lab. And then we divide them in 6 aquariums randomly. And the 6 aquariums were super temperature treatment. So we increased the temperature, and after the fish arrived to the target temperature, we let them be just there for 3 weeks. So the exposure time was 3 weeks. And after the 3 weeks, we started the testing in itself. So for the escape response, we use this setting. We put the fish on arena, and then we dropped a stimulus, and we recorded the fish behavior to then later analyze the videos. And we were looking for the fish non-locomotor variables. So if they did a startle response versus if they did not respond. When they did a startle response, if it was a fast start or if they were just bending. If they did a fast start, if it was like a C start or an S start. And then we also looked if the fish moved away or towards the stimulus and how much it took them to respond to the stimulus. After that, we also looked at the locomotor variables, so the escape distance they traveled and the maximum speed they achieved. After the escape response, we looked at the feeding rates. So for the feeding rates, fish were fed a fixed number of prey. The prey was live Artemia. And then the number of strikes was counted for 90 seconds. And a strike was considered every time the fish opened the mouth and moved the head towards the prey. Finally, we looked at the routine metabolic rate. We put the fish in these chambers and let them acclimate for an hour with the water running through. And then we close the water and start measuring for another hour their oxygen consumption. And what we find was really interesting. So for the escape response, neither of the non-locomotor variables, like the fast start, the no response, the direction they took, none of that was statistically different. But when we look to the locomotor variables, we saw that the escape distance was smaller in the fish, was significantly smaller on fish at 18 degrees, as well as the maximum speed they attained was also less than the speed they had in the controlled temperature, the 15 degrees temperature. When we looked at the feeding rates, we also realized that fish were consuming more food at the 18 degrees temperature compared to the 15 degrees, the controlled temperature. However, when we look at the metabolic rate, we saw that both high temperatures, so 18 and 21 degrees, increase their routine metabolic rate. As it is expected, usually when the temperature increases, the metabolism increases as well. And so when we stop to think and try to make sense of all of this, we really were surprised and thought this was interesting, because in one side, at 18 degrees, so within fish range of variability, fish decreased their speed and the distance they traveled, but they increased their feeding rates. But when we looked at the fish that were in higher temperatures outside their range of variability, the speed and the distance they travel was statistically the same as it was in the controlled temperatures, as well as the feeding rates. So, but when we look at the routine metabolic rate, it was higher at 18 and 21 degrees. So the metabolism increased, the oxygen consumption increased, and the fish under the 2 elevated temperatures both increased their metabolism, but how this increase translated into behavior was different. So the fish at 18 degrees increased their feeding, probably to compensate the increased metabolic demands, and decreased the activity, probably to conserve energy. But when fish were exposed to temperatures outside their variability range, they appear to disregard the conservation of energy, maintaining their activity as if they were in temperatures, in the environmental temperatures, but also didn't compensate the increased energetic demands with the increase of food consumption. So, what we saw here is this type of behavior can compromise mechanisms and on fish and impact processes, for example, like reproduction and predator-prey interactions. And what we saw with this experiment was that the project future scenarios of ocean warming that are expected to surpass the species variability will probably compromise fish acclimation responses, and this will affect their fitness and at long term, probably their survival. So yeah, so this was what we were able to gather from our 1-month experiment. And yeah, thank you all for listening, and to my supervisors and the technicians that help us. Thank you very much. If you have any questions.
Thank you, Anna. Any questions? So how does this translate into, you know, climate change or a wider ecology of this species, do you think?
Well, what we are looking, and this is part of my thesis actually, because I also studied the population in Portugal. That is the same it's the same species, not the same population, of course. Is that on the population on the fjord is probably less likely to acclimate to elevated temperatures, probably because it's a different habitat, and they also are acclimated to colder temperatures, because the temperatures in the fjord can go from 3 degrees to 18. So they probably their acclimation is more towards the colder side than the warmer side. And what you see is that species that are more south, so our, for example, our species, no, populations like ours, are able to acclimate to higher temperatures, continue to decrease their activities to maintain energy, and this are not. So I think yes,
I wonder a different kind of experiment. So what you have there in that experiment is a fish that has stayed at a lowish, more or less constant temperature, and then you put them at a warmer temperature. If you had exposed these animals to variable higher temperatures, so you know, once a while they could reach higher temperatures, I wonder if they would adapt better.
We did also an experiment in Christineberg, also with the sample, but a few years prior, where we did that. So we put the fish, we'll let the water just flow from the fjord, and then the temperature would increase 3 degrees more, whatever the water from the fjord came. So it was always varying, and it was also varying on the high temperature aquarius. And we saw that the ones in high temperature, once they surpassed this range of variability they have, they stop reproducing. So yeah, so we did that just with reproduction. We didn't test other things, but we saw that they stop reproducing.
But yeah, reproduction is quite a sensitive,
Yeah,
sensitive parameter. Okay, well, any other questions? Well, if not, I think we've finished for today. Thank you very much, Anna.
Thank you.